Hello, sir.
Good afternoon, madam.
Good afternoon.
Shall we proceed, madam?
Oh, yes, sir.
Yeah. Today's topic,
Today's topic is about immunology.
immunology.
Yes, sir.
Yeah, please, madam. Please proceed.
So, since the topic of immunology is very vast, so
Ah.
I think I will cover today only the basics of immunity.
Yeah,
In
yeah.
the
We
next
can
section,
we
I
can
will
make as a
continue
part
with,
one.
part one.
Yeah.
Okay. Hello, everyone. A warm welcome to today's class at Tawhid Foundation for Allied Healthcare Sciences, Bangalore. So in today's session, we'll be focusing on a very important concept in microbiology, that is immunology. So immunology is an essential field in both microbiology and healthcare, as it helps us understand how our bodies defend themselves against disease-causing microorganisms. So, introduction to immunity. The term immunity is derived from Latin word which is immunitas, which means freedom from disease. It is defined as the resistance offered by the host against microorganisms or any foreign substances. Immunity can be broadly classified into two types. First is innate immunity. Second is acquired immunity. Innate immunity present right from the birth, while acquired or adaptive immunity is acquired during the course of life. Classification of immunity. The diagram breaks immunity down into two primary types. First is innate, that is non-specific or natural immunity. Second is acquired, that is specific immunity. So first we'll start with innate immunity. This is the body's first line of defense and does not require previous exposure to the pathogen. It acts immediately or within hours of encountering an agent. Key components include skin and secretion. So, skin and secretions are physical and chemical barriers. So, saliva tears stomach acid that prevent entry of pathogen in these physical and chemical barriers. Phagocytes and REC. REC is reticuloendothelial cells, that is white blood cells, for example, neutrophils, macrophages that engulfs and destroy pathogens. Third is inflammation. It is localized response to infection or injury that results in redness, heat, swelling, and pain, which aids in defense and healing. NK cells, that is natural killer cells, lymphocytes that destroy virus infected cells and tumor cells is included in NK cells. Interferon, it is a protein released by cells in response to viral infection, which helps protect other cells from being infected. Complement, it is a group of proteins in plasma that helps antibodies and phagocytotic cells clear microbes and damaged cells. Second is acquired or specific immunity. So these develops after exposure to a specific antigen and is characterized by specificity and memory. It is further divided into humoral immunity and cell-mediated immunity. So humoral immunity in this B cells, that is lymphocytes that produce antibodies. B memory cell remembers past infections for a faster response upon re-exposure. Plasma membrane produce antibodies, that is immunoglobulin. So immunoglobulins, there are 5 types of immunoglobulin: IgM, IgG, IgA, IgD and IgE. Further, it is divided to cell mediated. So cell mediated immunity contains T cells. T cells is specialized lymphocytes that do not produce antibodies but help directly attack infected or abnormal cells. T helper cells regulate immune responses, whereas T killer cells, that is cytotoxic T cells, destroy infected or cancerous cells. T suppressor cells inhibit immune responses to prevent overactivity, whereas T memory cells remember past antigen exposure for a quicker response in future. Innate immunity. So, innate immunity is the inborn resistance against infection that an individual possesses right from the birth due to his genetic or constitutional makeup. So, it is immediate, that is first line of defense. Properties of innate immunity. First, acts in minutes. Innate immunity is the first line of defense against infections, occurs immediately after the microbial entry. Second is microbial exposure is not required. So here innate immunity is independent of prior exposure to the microbes present even before the first entry of the microorganisms. Third, diversity is limited. Innate immunity is active only against a limited time of antigens and is constant to acquired immunity, which is more varied and involves specialized immune response. Fourth is non-specific. Here, cells of innate immunity are non-specific in their action, can be directed against any microbial antigens. Last is no memory. Innate immunity does not have a memory component, so response to a repeat infection is identical to the primary response. Levels of innate immunity. First is species immunity. So, species immunity is the innate immunity towards a microbe exhibited by all members of a given species. For example, frogs are resistant to Bacillus anthracis while toads are susceptible. Second is racial immunity. Here, sometimes innate immunity is confined to a particular race, may be absent in another communities. For example, Negroes of America are more susceptible to tuberculosis than the whites, which is albinos. Individuals' immunity refers to the antimicrobial defense mechanism that are confined to a particular individual, may not be exhibited by others. Factors affecting innate immunity. So there are several factors determine the degree of innate immunity exhibited by the host, such as age. Certain infections are common in a particular age. For example, congenital infection like rubella is common in fetal life. Chickenpox and measles occurs in children, whereas urinary tract infection is common in adults. Second is hormones. Certain hormonal disorders, for example, diabetes mellitus, or patients on hormone therapy, for example, corticosteroids, are at increased risk of developing various infections. Third is nutrition. Malnutrition suppresses the host immunity, thereby predisposes to various infections. So, differences between innate and acquired immunity. Innate immunity is resistance to infection that an individual possesses from birth, whereas acquired or adaptive immunity is resistant to infection that an individual acquired during his lifetime. Innate immunity response occurs in minutes, whereas acquired occurs in days. Innate immunity's prior exposure to the antigen is not required, whereas in acquired, following the antigen exposure develops. In innate immunity, diversity is limited, acts through a restricted sets of reaction, whereas in acquired, more varied and specialized responses occurs. Immunological memory responses are absent in innate immunity, and immunological memory responses are present in acquired immunity. Microbial and antigen. Innate immunity develops against antigen that are shared by many microbes, are called as microbes associated molecular patterns, that is MAMP, whereas in acquired immunity, microbial antigen, acquired immunity develops against antigen that are specific for each microbes. Host cell receptors of innate immunity, called pattern recognition receptors, are non-specific, for example, toll-like receptors, and host cell receptors are specific, for example, T cell receptors and B cell immunoglobulin receptors. These are the components of innate immunity, and these are the components of acquired immunity. So we'll start with components of innate immunity. Anatomical barriers. First, what is components of innate immunity? There are several mediators of innate immunity. They are the cells, the proteins, the antibodies, the antibiotics, the antibiotics, the antibodies, 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 the antibodies They exert antimicrobial activities by various mechanisms as described in all this. So, more so, many mediators are not purely part of innate immunity. They often act as a bridge between innate and acquired immunity. So, example is complements and macrophages. We'll start with anatomical barriers. Anatomical barriers such as skin and mucosal surfaces have a spectrum of antimicrobial activities. Physiological barriers that contribute to the innate immunity are the body temperature, pH, and various soluble secretory products of mucosa. Phagocytes such as neutrophils, macrophages, including monocytes, are the main components of innate immunity. They are rapidly recruited to the infection site. Phagocytosis involves three sequential steps. First is engulfment of microbes and subsequent hosting in phagosomes. Second is fusion of lysosomes with phagosome to form phagolysosome. And last is microbial killing. Fourth is natural killer cells. It includes mast cells and dendritic cells. So natural killer cells, they are classes of lymphocytes that kill virus infected cells and tumor cells. So NK cell mediated mechanism of killing microbes, I'll explain later. Other rare classes of lymphocytes, T and B lymphocytes, are the chief mediators of acquired immunity. However, there are several rare types of lymphocytes that share the features of both acquired and innate immunity. So T cell, also called as intraepithelial lymphocyte, they are present in epithelial lining of skin and mucosa. NKT cells, they are present in epithelium and lymphoid organs. Mast cells, they are present lining the respiratory and other mucosa. They are activated by microbial products binding to toll-like receptors or by IgE antibody-dependent mechanism. So following which they release abundant cytoplasmic granule rich in histamine, prostaglandins, and cytokinins that initiate inflammation and proteolytic enzymes that results in killing of bacteria. Dendritic cells, they respond to microbes by producing numerous cytokines that initiate inflammation. They also serve as vehicle in transporting the antigen from the skin and mucosal sites to lymph nodes where they are present the antigens to T cells. Hence, dendritic cells serve as a bridge between the innate and acquired immunity. Complement pathway, alternate and mannose building pathways are the chief mediators of innate immunity. Alternate complement pathway is activated in response to bacterial endotoxin, whereas the mannose binding pathway is stimulated by mannose carbohydrate residues on bacterial surfaces. So, following activation, the complement mediate various biological functions such as lysis of the target microbes, stimulate inflammation, and stimulate acquired immunity. Last is fever and inflammatory responses. Inflammation is defined as the biological response of vascular tissues to harmful stimuli such as microorganisms or other foreign substances. The major events that take place during an inflammatory response following a microbial entries, that is vasodilation due to the release of vasoactive substances from the damaged tissue, leakage of plasma proteins through blood vessels, recruitment of phagocytes, that is neutrophils, to the sites of inflammation. Phagocytes undergo the following step, that is margination, second is rolling, endothelium, third is extravasation. Last is engulfment of microbes and
Hello,
materials by the phagocytes and destruction of the microbes.
madam. Voice is, I think, a little bit. It is slow. I think. Just
All right,
make
sir,
sure
I'll make
net
sure.
connection is okay, na?
Okay, sir, I'll just check.
Okay.
Hello, now is it is proper?
Yeah, madam. Please proceed.
All right. So, anatomical and physiological barriers. There are some features like skin, mucous membrane, cilia, saliva, tears, gastric juice, bile salts, and normal flora. Skin, mucosal, cilia, saliva, tears, gastric juice, all this comes in anatomical barrier, and physiological barrier, uh, last bile salts and normal flora. So skin mechanically prevents entry of microbes, produces sebum containing antimicrobial peptides and fatty acids, which helps in killing of microbes by lymphocytes. Mucosal membranes prevents entry of microbes mechanically and by producing mucus, which entraps microbes. Cilia present in the lower respiratory tract propel the microbe outside. Saliva, enzymes in saliva damage the cell wall and cell membrane of bacteria. Tears containing lysozymes that destroy the peptidoglycan layer in bacterial cell wall. Gastric juice, HCl kills microbes by its low pH. Bile salts interfere with bacterial cell membrane. Last, normal flora, intestinal and respiratory mucosa are lined by normal flora, which helps to kill bacteria. These are the examples of anatomical and physiological barriers along with functions. Mechanism of innate immunity. Receptor interaction. Following the exposure of microorganisms, several mediators of innate immunity are recruited to the site of infection. So the first step that takes place is attachment, which involves binding of the surface molecule of microorganism to the receptors of cells of innate immunity. Second is microbial surface molecules, which is MAMPs. They are the repeating patterns of conserved molecules which are common to most microbial surfaces called microbes associated molecular patterns, MAMPs. Example of MAMPs include peptidoglycan, lipopolysaccharides, teichoic acid, and lipoproteins present on bacterial surfaces. Third is pattern recognition receptors, PRRS. So these are the molecules present on the surface of host cell. For examples, phagocytes that recognize MAMPs, they are generally conserved regions encoded by germline genes. Toll-like receptors, TLRs signaling are classical examples of pattern recognition receptors. So signals generated following binding of TLRs to MAMPs activate transcription factors that stimulate expression of genes encoding cytokines and enzymes, which are involved in several antimicrobial activities of cells of innate immunity. Toll-like receptors, they are so named because they are similar to toll receptors present in the fruit fly Drosophila, where it is the main receptor for inducing innate immunity. Total, there are 13 types of toll receptors, TLR 1 to 13, recognized so far, which bind to particular MAMP molecule on molecular surfaces. So, out of which important one are TLR2, which is peptidoglycan, TLR4, that is binds to LPS of gram-negative bacteria, TLR5 binds to flagella of bacteria. Major events in anti-inflammatory responses. So this diagram illustrates the major events in an anti-inflammatory response after tissue damage, specifically when bacteria evade through a break in the skin. So first, tissue damage and release of mediators. When tissue is damaged, example, bacteria enter through a wound, damaged cell, release chemical mediators such as histamine and cytokines. So these mediators signal nearby blood vessels to increase capillary permeability, allowing immune cells and substances to leave the blood vessels more easily. Leakage of fluid and cells. The increased permeability enables fluid and immune cells to leak out of the capillaries into the surrounding tissue. This leads to margination, where immune cells like phagocytes move towards the margin of the blood vessel to prepare for existing into the tissue. Migration of phagocytes. Phagocytes such as neutrophils and macrophages migrate from the bloodstream into the site of injury. This process is called extravasation, movement of immune cells out of the capillary into the tissue. So these cells are directed into the infection by chemical signals, chemotos, with that is chemotoxis. Fourth is phagocytes and exudate destroy bacteria. Once in the damaged tissue, phagocytes engulf and destroy bacteria and other harmful microorganisms. So visual breakdown, tissue damage, top broken skin allows bacteria to enter. Release of mediators triggers an inflammatory reaction. Exude form as fluid and cells leave the capillaries. Fourth, phagocytes migrate out of the blood cells into the tissue. And last, phagocytes and exudate work together to destroy bacteria and protect tissues. So these are the major events in an inflammatory responses. Acute phase reaction proteins, APRS. They are the proteins synthesized by liver at steady concentration, but the synthesis either increases or decreases exponentially during acute inflammatory conditions. Through liver is the primary site. APRS can also be synthesized by various other cells such as endothelial cell, fibroblast, monocytes, and epidocytes. Positive APIRS, they are the proteins whose level increases during acute inflammation. Example includes serum, C-reactive proteins, complement proteins, coagulation protein, proteinase inhibitor. These are the examples. Negative APIRS, they are the proteins whose level are decreased during acute inflammation, thus creating a negative feedback that stimulates the liver to produce positive APIRS. Role of APRs, they have a wide range of activities that contribute to the host defense. APRs have various antimicrobial and anti-inflammatory activities, that is, complement factors. Metal binding proteins can chelate various metals such as iron, copper, etc., making them unavailable for the bacteria. So main role is antimicrobial and anti-inflammatory properties of APRs. C-reactive protein. C-reactive protein is an example of APR that rise in acute inflammatory conditions, including bacterial infections. It belongs to beta globulin family. Markers of acute inflammations: normal less than 0.2 mg per dl rises in infections and trauma. Detection is latex agglutination test, HC-CRP. So CRP can be detected by precipitation method using C carbohydrate antigen, not in use now. Latex passive agglutination test using latex particle. It is the most widely used method employed worldwide. Detection limit of CRP by latex agglutination test is 0.6 mg per dl. So highly sensitive CRP has HSCRP test. Minute quantities of CRP can be detected by various methods, enzyme immunoassays, etc. Characteristics of acquired immunity. So, response in days. Acquired immunity involves activation of T and B cells against the microbial antigens, which takes several days to weeks to develop the following microbial entry. Requires prior exposure. Acquired immunity develops only after the exposure to the microbes. Highly specificity, specific acquired immunity is highly specific, directed against specific antigens that are unique to the microbes. Memory present, Acquired immunity does not have a memory component, so a proportion of T and B cell become memory cell following primary contact of the microbe, which play an important role when the microbe is encountered subsequently. Wide diversity. Acquired immunity, though takes time to develop, is active against a wide range of antigens. Types of acquired immunity. Acquired immunity can be classified into two ways based on mechanism, active and passive, based on source, natural and artificial. Active immunity. Active immunity is the resistance developed by an individual toward an antigenic stimulus. So here the host immune system is actively involved in response to the antigenic stimulus, leading to the production of immunologically active T cells, B cells, and production of specific antibodies. Active immunity naturally or artificially. So, natural active immunity occurs following an exposure to microbial infection, for example, measles and virus infection. Artificial active immunity develops following exposure to an immunogen by vaccination, example, measles vaccines. So, vaccines are discussed later. I'll explain later. Passive immunity. Passive immunity is defined as the resistance that is transferred passively to a host in a ready-made form without active participation of the host immune system. Passive immunity can also be induced naturally or artificially. Natural passive immunity involves the IgG antibody transfers from mother to fetus across the placenta. Artificial passive immunity develops following ready-made transfer of commercially prepared immunoglobulin, example, rabies immunoglobulin. Passive immunity plays a very important role in immunodeficient individual act as host immune apparatus is not effective and post-exposure prophylaxis when an immediate effect is warranted. Secondary immune response. Primary and secondary immune response. So here in this diagram, I'll explain. These diagrams display the primary and secondary immune response, how the body responds to an antigen for the first and subsequent time. So primary immune response is first antigen exposure. Its lag phase is 4 to 7 days. Antibodies mainly involved IgM. Secondary immune response is subsequent exposure. It is rapid, powerful IgG dominant immunoglobulin basis of booster doses. So in this diagram, primary antigen exposure, lag phase. Upon the first exposure to an antigen that is foreign substance, there is a delay called the lag phase before an immune response is detected. So here immune cell recognize the antigen and prepare to respond it. IgM low titer. After the lag phase, the body produces IgM antibodies that is immunoglobulin M. So the graph shows a low titer, low concentration of IgM in the blood, which helps fight the infection during this initial exposure. Negative phase. After the primary response, if the antigen is cleared, antibody levels drop. This decrease is referred to as the negative phase. Secondary antigen exposure. Upon re-exposure, secondary antigenic exposure to the same antigen, the immune system reacts much more quickly and strongly thanks to memory cells generated during the first exposure. So, IgG high titer. Now the body produces IgG antibodies, that is immunoglobulin G, in higher titer, that is higher concentration, and much faster than before. IgG provides a more efficient, rapid, and long-lasting protection against the antigen. So this primary immune response. Latent or lag phase, active immunity develops only after a latent period following the antigen exposure, which correspond to the time required for the host immune apparatus to become active. And secondary immune response, when the same antigen exposures occur subsequently, the event which takes was shown in that figure. These are the active versus passive immunity. Active immunity produced actively by host immune system. Passive immunity, immunoglobulins received passively. It is active immunity is induced by infection, which is natural, vaccination, artificial. Passive immunity acquired by mother to fetus, IgG, transfer naturally. Ready-made antibody transfer, that is artificial. Active immunity is not long lasting. Passive immunity lasts for short period of time. Active immunity is lag period it present in active immunity, no lag period present in passive immunity. Memory is present in activity, whereas no memory present in passive immunity. Booster doses are useful in this active immunity, whereas subsequent doses lessen its activity effective. Negative phase may occur in active immunity, whereas no negative phase occurs in passive immunity. It is not useful in immunodeficiency individuals for active immunity. It is useful in immunodeficient individuals for passive immunity. Bridges between innate and acquired immunity. The innate and acquired immunity do not work independently. Rather, they function in a highly interactive and collaborative manner, increasing each other's efficiency and producing a combined response, which is more effective than either branch could produce by itself. So, certain immune components play important roles in both types of immunity and are considered as bridges between innate and acquired immunity. So I'll give examples, that is macrophages and dendritic cells belong to innate immune system, but as antigen presenting cell they present the antigenic peptides to T cells, more so cytokinins secreted from macrophages, that is interleukin 1, are also involved in T cell activation. ADCC, antibody dependent cell mediated cytotoxicity, is also a type of cell mediated immune response. Complement systems, classical versus alternate. So classical pathway are also part of both innate and adaptive immunity that destroy the target cell which are coated with specific antibodies. However, alternate and mannose binding pathway do not take help of antibodies. Cytokines secreted from cells of innate immunity can activate cells of adaptive immunity and vice versa. Local or mucosal immunity. So local or mucosal immunity is the immune response that is active at the mucosal surfaces such as intestinal or respiratory or genitourinary mucosa. It is usually mediated by type of IgA antibody called secretory IgA, which prevents the entry of microbes at the local site itself. Local immunity can only be induced by natural infection or by live vaccination, but not by killed vaccines. So, for examples, following administration of live oral polio, OPV, or following infection with poliovirus, secretory IgA antibodies are synthesized and coated on intestinal mucosa, which prevents subsequent poliovirus infections. Such immunity does not develop following injectable killed polio vaccines. Herd immunity. Herd immunity is defined as the overall immunity of a community or herd towards a pathogen. Herd immunity plays a vital role in preventing epidemic diseases. If the herd immunity is good, that means large population of the community are immune towards a pathogen. So hence epidemics are less likely to occur and eradication of the disease may be possible. Elements that contribute to create a strong herd immunity are occurrence, clinical and subclinical cases, ongoing immunization program, herd structure, type of pathogen. Herd community may not be strong in a community against all the pathogen. So herd community develops the following: effective vaccination against some diseases like diphtheria and pertussis vaccine, measles, mumps and rubella vaccine, polio, OPV vaccine, and smallpox vaccine. Last is adaptive immunity. Adaptive immunity is a special type of cell mediated immune response which develops following injection of immunologically competent T lymphocyte known as transfer factor. It is useful for treatment when the CMI is low, that is in lepromatous leprosis. Summary and key takeaways. So, in this whole session, we learned about immunity, that is innate, immediate, non-specific immunity, and acquired, specialized, specific memory. Active and passive forms of immunity. Barriers, phagocytes, complement, all these three things plays vital roles in immunity. Vaccination builds adaptive immunity and herd protection. Questions? So these are the 10 important questions for 3 marks, which is very basic. Define first, define immunity. Second, what is innate immunity? Third, give 2 examples of each. Fourth, list 3 differences between innate and acquired immunity. Define species immunity with 1 examples. What is the role of natural killer NK cells, dendritic cells? Define C-reactive protein, CRP, and mention its normal level. Define active immunity with an example. Define passive immunity with an example. What is primary immune response? Define herd immunity with 1 example. 5th mark questions. First, define immunity and differentiate between innate and acquired immunity. Explain the properties and components of innate immunity. Describe the factors affecting innate immunity. Write short notes on species, racial, and individual immunity. Explain the mechanism of phagocytosis in innate immunity. Define acquired immunity and explain its active and passive types with examples of each. Differentiate between primary versus secondary immune responses with a neat label diagram. Write a note on acute phase reactant proteins, that is APR, with suitable example. Explain herd immunity and its importance in disease prevention. And last is describe the bridges between innate and acquired immunity, why it is important. So that's all for today's class.
Yeah, thank you so much, madam. Nicely have
Thanks.
explained about immunity.
Yes, sir. It is actually a wide topic, so very
Yeah, yeah.
confusing also and quite tricky also.
Yeah, yeah. So, but you have presented in a concise manner.
Yeah, so that is why I just took
Yeah,
few
yeah,
parts of only immunology.
yeah,
In next
yeah,
session, I can explain
yeah.
antigen, antibody, hypersensitivity, everything in
Yes, we
detail.
can continue further. We can continue.
E for?
So, as per, I think, introduction part, I think you have
Yes,
covered.
yes, sir.
In detail, we can cover in next sessions.
Next session. Yes, sir.
Yeah. Thank you, madam. Thank you so much.
Thank you,
Taking your
sir.
time,
Thank you, sir.
that too 4 to 5 p.m.
Yes.
Thank you, madam. Thank you so much.
Thank you, sir.
Shall we wind up, madam?
Yes, sir, sure.